since 1981, when the first case of HIV was reported in the United States. A vaccine has remained beyond reach for a few reasons, including the rapid mutation of the virus, which leads to treatment resistance. Antibodies that are effective against multiple HIV-1 strains, known as broadly neutralizing antibodies, are a promising treatment option, but they still need to be stronger and better designed to maintain their efficacy against virus mutation. They can also take years to develop as human antibodies, which consist of heavy and light amino acid chains, cannot easily maneuver around the natural defenses the virus has set up to reach their target binding sites on the virus surface. Nanobodies, small fragments of antibodies, could offer a solution to this predicament. A potent antibody therapy derived from llamas was found to broadly neutralize numerous strains of HIV-1. Camelid animals, including camels, alpacas, and llamas, produce a specific type of antibody known as heavy-chain-only antibodies, which are the main source of nanobodies. Nanobodies are about 10 times smaller than conventional antibodies. They have an elongated shape and are good at binding smaller, often partially occluded sites on HIV-1 antigens, which may be inaccessible to classical human antibodies. Nanobodies neutralize HIV-1 antigens by binding to the envelope protein spike, which the virus uses to enter healthy host cells, thus shutting down infection. To obtain the nanobodies, the scientists first needed to induce the production of heavy-chain antibodies in a camelid animal. To do this, researchers injected a single llama with an HIV-1 antigen, the part of the virus that the immune system recognizes as invasive, prompting it to respond. The HIV-1 antigen they used was fixed in a closed conformation with well-exposed target sites, making the immune system more likely to detect the virus. In response, the llama, immunized 13 times over the course of 271 days, produced blood cells that generated heavy-chain-only antibodies from which nanobody genes could be isolated. These genes allowed the researchers to produce large quantities of the nanobodies in a controlled environment using a technique called phage display, in which the nanobody genes were inserted into viruses that infect bacteria called phages. These phages display the nanobodies on their surface, allowing them to bind HIV-1 antigens. Researchers used different HIV-1 protein antigens to select for phages that displayed antigen-binding nanobodies on their surface. Among the numerous nanobody candidates isolated from the llama's blood, the researchers identified those that were most effective against a range of HIV strains and had the strongest ability to neutralize the virus. Although the best nanobodies demonstrated promising results on their own, the researchers aimed for even better neutralization performance. The goal for HIV-1 neutralization is to achieve 100% neutralization at the lowest possible concentration. For a therapeutic, it is desirable to inhibit many, if not all, virus strains, enabling close to complete viral suppression in AIDS patients. Even though a single broadly neutralizing antibody or nanobody may be able to neutralize most or even all HIV-1 strains, new mutants will continue to be generated owing to the instability of the virus genome. Treatment with one antibody targeting a single site usually leads to the accumulation of virus mutants that are resistant to the antibody. Researchers engineered a human llama chimera or bispecific antibody that can bind to two of the virus-vulnerable sites simultaneously, making it much more difficult for the virus to evade neutralization. This two-pronged attack is possible owing to the different shapes and binding preferences of the llama nanobody and human antibody, which were linked together. The nanobody binds to a specific site known as the CD4 binding site, while the human antibody targets another site known as the V2 apex. Not only a very small concentration of the human llama antibody needed to neutralize the HIV-1, but it neutralized more than 95% of 208 HIV-1 strains simultaneously, making it 10 times more potent than the human llama antibody the researchers previously engineered. Although these results are promising, more practical aspects need to be taken into account, including the antibody's half-life or the time required for its concentration to diminish by half after injection in the human body. The researchers immunized only one llama. At this stage, it's unclear if immunizing more llamas would lead to broader HIV-1 neutralization or if other animals could produce more potent nanobodies. Despite the remaining obstacles, the newly developed antibody represents progress in the now decades-long effort to develop an HIV-1 treatment that can effectively inhibit all strains of the virus. Thank you for watching. If you've enjoyed our videos, subscribe to our channel for there's more to come.